It's really great to have you all here tonight. As you know, yesterday Steve Meyer and Doug Axe were out at Biola, and there was a great event, I understand, at Biola University last night. I don't know what, well, I do know part of what you did this morning, but it didn't have to do with this, but other things. And we had a great lunch today with some folks who are looking to start a chapter of the Center for Science and Culture right here in Southern California. Some of them are with us tonight. And then tonight we get to hear my friend Steve Meyer tell us what's happening in the research that has grown out of a design thesis. I've got the official bio, and when I read it, I thought, I do need to tell you this stuff. I usually tell the story of how, and some of you heard it earlier today, but I don't know what year it was. It must have been 91 or 92. A person I knew, and some of you may know, named Celia Yates, who was a book agent for Christian books. And he asked Howard, he was very important because he talked Howard, well, he talked the person who worked for Howard out of forcing me to sign a prenuptial agreement, which I would not have done. You know, you want a prenuptial agreement? Fine. I was looking for someone when I met you, and I'll be looking for someone from now on, because I'm not signing any prenuptial agreements. At any rate, because I'm not presuming divorce. I'm in this for life. Right, Howard? Yeah. Murder, yes. Divorce, never. Okay. Anyway, so I loved Celia Yates. Let's just put it that way. At any rate, he calls us up and says, there's somebody I'd like you to meet. And so, yeah, Agatha, what did you say?
Agatha Christie made a whole career off that philosophy.
Yeah. If you didn't hear it, he said Agatha Christie made a whole career off that philosophy. So there you are. Anyway, I can't take you anywhere except you own it. So what am I supposed to do? Anyway, we met. The gentleman in question was a man named Philip Johnson, who was a professor at the University of California School of Law, Bolt Hall. He had clerked for one Supreme Court Justice, Earl Warren, you've heard of him, and he had also clerked for state Supreme Court Justice, one or two here in California, I think. He was 28 years old when he got, I think, tenure at Cal Berkeley Bolt Hall. When I was 28 years old, I was on unemployment insurance, so he clearly had something that he had figured out. So at any rate, Phil Johnson, he had gotten a sabbatical. He taught evidence in tort law, and he had a sabbatical in London, and he walked by a bookstore of science books, and he was reading all the Darwin stuff, and when he read it, for some reason he just got interested, and he said none of this stuff would hold up as evidence in a court of law. And he wrote a book called Darwin on Trial, and it was popular in Christian circles. It wasn't so popular anywhere else. And Celia wanted us to meet him. And there's a long story. I'll spare you both Howard's and my life stories because we would never get to hear Steve. But at any rate, this resonated with each of us for very particular reasons in our own biographies. And so the upshot was we said, well, what can we do? Well, Phil Johnson didn't need any money. I mean, he has, tenure at Cal Berkeley. He's fine. And so we didn't need money. But he said, I've never gotten all these guys together. And for some people, Phil Johnson was the first person they ever got an email from. He was one of the very first people on CompuServe, for those of you who are old enough to remember CompuServe. At any rate, he said, I've identified all these young scientists who have serious questions about the Darwinian paradigm, but they've never gotten together. And so he said, if you could put up the money for them to get together, that'd be great. And it'll be 25 years ago this summer that happened up here in Northern California. And so that's my origin story. And Steve Meyer had met Phil Johnson when Phil was in London. And I learned today that the meeting was set up by someone who wanted Steve to talk Phil out of being so interested in this science stuff. It was a friend from California who knew Phil and said, he's just gone off on this thing. It's just crazy. You've got to talk him out of it. Well, apparently that didn't work. So anyway, here we are. Steve Meyer, since then, has done many impressive things. And I've got to give you the goods here, and I've got to have it because the list is so long, even I can't remember. He got his doctorate in the philosophy of science from the University of Cambridge. Before that, he had worked as a geophysicist, and he became a college professor. He now directs the Discovery Institute's Center for Science and Culture. He's the author of the New York Times bestseller, Darwin's Doubt, The Explosive Origin of Animal Life and the Case for Intelligent Design. And then he is the author of Signature in the Cell, which was a Times London Literary Supplement Book of the Year. That started with an article that was published in the Proceedings of the Biological something of Washington, D.C., published by the Smithsonian. And the editor who published that lost his position as a result of that move. And so the upshot, there was quite a brouhaha about that. I think even a congressional inquiry into why he lost his job. And so Steve Meyer has caused trouble. He's been featured on national television. The list is really kind of too long. The Jim Lehrer NewsHour, CBS Sunday Morning, NBC Nightly News, ABC World News, Good Morning America, Nightline, Fox News Live, Paul is on now. It goes on. And so he's going to tell us tonight what the latest stuff is. How's that for a scientific term? Stuff. Steve Meyer. And you go up here.
Yeah, when I was growing up, my mom often said, Stephen, we're all right here. So I'm not sure I need the mic, but I'll try to modulate the voice. A little bit of a tall order tonight. I've been asked to talk about the new cutting-edge research that the theory of intelligent design is inspiring, and I'm excited to do that. I also realize there's a lot of people who are just getting acquainted with some of these ideas, so I'm going to do two things tonight, and I'm going to start with a little bit of an overview of the core ID intelligent design arguments, and then I'm going to show how those very arguments have inspired further research. So for the first part won't be new for some, but it'll be necessary background to where we're going. I'm also going to bring Doug Axe up a little bit later, and we'll do we've got some really neat animations to show of some of the things that are going on inside the cell and how that's leading to even to some medical breakthroughs. And we're just going to talk a little bit interactively about some of those things at the very end. So we'll try to keep this lively. With your indulgence, I was scheduled to go 45, but I think this will be a better talk if I'm closer to 50 to 55, if that's all right, because there's just some really cool stuff to share, and I do want to give the background for people who haven't been exposed to intelligent design before. Okay, we begin. November of 2016, Doug Axe and I and 18 other scientists who favor the theory of intelligent design attended the meeting of the Royal Society in London. The Royal Society is the oldest and most august scientific body in the world. It goes back to the 17th century. Sir Isaac Newton was one of its presidents. They had the portraits of all these heroes of science. But this year, the meeting in late 16 was called by leading evolutionary biologists who realized that neo-Darwinism, the standard textbook theory of evolution that we all learn in high school and college biology courses, is in serious trouble. In fact, the opening talk of the conference was by a very prominent Austrian evolutionary biologist named Gerd Müller, whose talk was titled The Explanatory Deficits of the Modern Synthesis, which is a jargon term for neo-Darwinism. His talk described most poignantly the inability of the mutation natural selection mechanism to generate genuine novelty. He said the mechanism lacks creativity. It doesn't explain the origin of major morphological innovations, the origin of new form. Pretty interesting, because you think back to Darwin's theory, when he explained the origin of new species, he was explaining the origin of new forms of life. So you have leading modern Darwinists saying, theory's not getting it done anymore. The mutation selection mechanism lacks creative power to explain the most important things in the history of life. Does a good job of explaining the small-scale variations, the little finch beaks that get bigger and smaller in response to weather patterns, The peppered moths, whose coloration turned dark and light and dark again in response to varying levels of industrial pollution. No problem with that. But explaining where birds and insects come from in the first place, or animal body plans, or still less, the origin of the very first life, we need a new theory, said these gathered evolutionary biologists. That's why they were there. Now, they were exploring many other ideas, not the theory of intelligent design that I'll talk about tonight. They were working within a naturalistic or materialistic framework. So they looked at a lot of other evolutionary models, evolutionary mechanisms. But at the end of the conference, one of the people who had been instrumental in calling the conference together, a science writer named Susan Mazur, who's written about the post-Darwinian evolutionary biologists, a group famously known as the Altenberg 16, said that sadly the conference was characterized by a lack of momentousness. There was many very trenchant critiques of the established theory, but no real breakthroughs on new mechanisms that would provide some insight into where the creativity must have come from, where the innovation must have come from. Now, we didn't really, that is, we in the intelligent design research community, or movement as we're sometimes called, didn't really need the Royal Society conference to inspire us to think that there was another approach that was needed to look at life in a different and more fruitful way. But it certainly didn't help to have leading evolutionary biologists confirming many of our very same critiques. And so what I'd like to talk about tonight is the theory of intelligent design, a little bit of the background on the theory, some of its core arguments, and then talk about the future we see for biology when biology is informed by the idea that a designing agent played a role in the origin of new forms of life, that life is a design system rather than a system that arose through unguided and undirected processes. So it might be good to start first with a definition of the theory of intelligent design. I'll be on a talk show tomorrow, Adam Carolla, a new guy I haven't talked to before. Anyway, he did a pre-interview, his guy did a pre-interview with me today and wanted a definition. Well, here's a definition. The theory of intelligent design says that there are key features of living systems, and the universe for that matter, but we're going to talk mainly about biology tonight, not physics and cosmology. There are key features of living systems that can be best explained by the activity of a designing intelligence, not an undirected natural process such as natural selection, acting on random variation or random mutations. And a corollary to that is that living systems look designed because they really were designed. Now, that seems almost like common sense, but it's radical in modern biology because the standard Darwinian view and modern neo-Darwinian view is that biology, in the words of Richard Dawkins, is the study of complicated things that give the appearance of having been designed for a purpose. Anyone identify the key word in that? It's the appearance, the illusion of design. Francis Crick, as Jim Townsend reminded me, has famously said that that biologists need to constantly remind themselves that what they're looking at evolved rather than they must constantly remind themselves that what they're looking at was not designed but rather evolved. So this is the Darwinian approach. Things look designed but they're not really designed. Why? Because there's an unguided, undirected process that can produce the appearance of design without itself being guided or directed in any way. If the appearance is an illusion, then whatever process produced it must be unguided and undirected. And that is what evolutionary biologists have long said about the natural selection random mutation mechanism that Darwin pioneered and which modern biologists have advanced. The problem is, as I mentioned, more and more biologists are recognizing that the mutation selection mechanism lacks creative power, and for some reasons that I'll discuss in a bit more detail in a bit. So the key question in biology is that precisely this. It's a question about design. It goes back to Darwin. It's right up to the present. Is the design real or is it imaginary? Is it illusory? Is it an illusion or is it real? Now, the folks in the intelligent design movement, the scientists in the intelligent design movement have been saying, we think it's real and we think there's some good reason to think so. Some reasons about which, or some evidence for design, there's now evidence of design that Darwin didn't know anything about. For example, on the inside living cells today, there are dozens of little nanomachines, tiny little machines, some motors, some walking motor proteins, some sliding clamps, little turbines. And one of those machines was made famous in a 1996 book by one of our colleagues, Michael Behe, Darwin's Black Box. Anyone heard of it? Okay. It sold over 300,000 copies, which for a science book is a monster bestseller. And it kind of announced to the world that there was this new way of looking at life called intelligent design. Now, Behe looked at, for example, a little miniature machine inside the cell wall of a bacterium called a flagellar motor. This is an animation still of the flagellar motor. You can see that it has a drive shaft, it has bushings, it has bearings, it has a U-joint, it has a rotor and a stator. It has all the parts of a rotary engine. In fact, that's what it actually is by all people who have studied it, the cell biologists. This is actually a beautiful X-ray picture of the actual system and the parts. We can actually really see what these guys are made of. Now, they've got all kinds of interesting design parameters with these machines. It's a rotary engine, it's water-cooled, it's driven by proton motive force, it's got 2 gears that go reverse and backwards. In some system, the rotary engine the rotor spins it up to 100,000 RPM and can reverse direction on a quarter of a turn. It's also hardwired into a signal transduction circuit that's giving the little bug information about where the concentrations of sugar are in the surrounding medium so that it can change directions and chase its food source down. I like to say this is the system is high-tech in low life. Little bacterium, but really sophisticated, stuff that looks like it's engineered. Now, Behe, of course, is well aware of the Darwinian perspective, that things just look designed, but he said, and the other cell biologists are saying the same thing, this looks like it was designed by a human engineer. But Behe said, okay, this looks designed, but this is a system that looks designed and really is best explained as the product of actual design, because according to natural selection, or according to Darwin's theory and modern neo-Darwinism, natural selection selects for functional advantage. But in a system like this that has many interlocking or interdependent parts, there's no functional advantage. The motor doesn't work until you get the whole suite, the whole complement of parts. So it's got 30 separate parts made of proteins, and the 29, 28, 27, 26, 25 part version of this performs no function. So there's no possibility of building this up gradually because those intermediate stages confer no functional advantage, therefore they won't be selected and passed on to the next generation, and therefore the evolutionary process will terminate when it encounters those non-functional configurations of matter or configurations of protein. So therefore, Behe says if the designer's substitute mechanism of natural selection can't explain the origin of this, maybe it really is designed. If it looks like a duck and quacks like a duck, maybe, just maybe, it actually is designed. Now, there's a more sophisticated rendering of that argument. He develops that in part. Others have amplified it. But he produced this monster best-selling book in the 1990s, advancing this first concept of intelligent design based on what he called the irreducible complexity of these miniature machines, these nanomachines in cells. And there are actually many others. Now, bringing us up to date, there was a counter-argument to Behe that came online very soon after, and especially at a famous court trial in Dover, Pennsylvania in 2004. And there has been in these little bacteria also another little molecular machine with fewer parts called a type 3 secretory system. And the type 3 secretory system secretes various products of what's going on inside the cell out of the cell wall. And this secretory system has many parts in common with the bacterial flagellar motor, in fact, the parts that are on the interior of the motor. And so the proponents of neo-Darwinism saw this and said, ha-ha, that's the precursor. That's the intermediate form that evolution used to build up to the full flagellar motor system. And this was put forward at the Dover trial as a definitive refutation of Behe and the idea of intelligent design. But it turns out that there's a problem that was concealed in this initial critique of Michael Behe. And that is that the genes that build the protein parts in this system are actually younger, they're younger than the genes that build the flagellar motor itself, if we go back to that system. And so what we actually have, and there's ways to assess that. Sorry, let me go find the picture from before. Going backwards. Yeah, here we go. So this is the flagellar motor. This type 3 secretory system is very similar to the parts on the inside, but it turns out that the genes that are necessary to build the proteins that make up this are younger rather than older than the genes that make up the type 3 secretory system. So it looks to be a devolutionary breakdown product of the aboriginal system. Not the precursor, but a product of devolution, not evolution, as the British would say. And there's an even deeper problem. And that is, in order to build a flagellar motor, there's a very precise sequence of parts that have to be put together in a precise order. You can't just throw all these protein parts together and just hope they'll self-assemble it. It's kind of like an airplane factory. There's a scope and sequence. You've got to have it in the right way. And that means that the genes that get turned on to build the different proteins have to get turned on in a very precise sequence. But that requires a whole host of regulatory genes and proteins that form a system, an information processing system, which is itself irreducibly complex. You remove any one of those proteins or genes, and you can't attempt to accomplish this assembly pathway. So the idea was, well, if we have this type 3 secretory system, then these other proteins would just get built willy-nilly, and then somehow they'd all get put together. But the only way all protein parts get put together into the more complex system is if there is an integrated information processing system that includes these regulatory genes and regulatory proteins. But that system itself has this tight functional integration of many parts such that if you remove any one, it doesn't function. So in other words, so to answer the argument from irreducible complexity, the critics had to invoke a process that was itself irreducibly complex. Now, the word on this hasn't gotten out very far, but we've popularized it, we've got some technical articles on it, but we've also popularized it in a new film called Revolutionaries, which is about the life of Michael Behe and the work and his work. So that's one of the key ID arguments. But keep this idea of irreducible complexity or the functional integration of parts in your mind, because that's going to be a key concept that we're going to use to generate a new perspective on biology and to generate some new testable models, especially testable hypotheses, especially as it relates to other nanomachines in the cell. Now, the second and perhaps more fundamental, and I think it is a more fundamental argument for intelligent design, is the argument from information, genetic information, which is essentially digital or typographic in the sense, and you'll see why in a minute. And this is I used to ask my students when I was teaching, if you want to give your computer a new function, what do you have to give it? Programming, right? Which is code or instructions, right? Well, this is the big discovery of modern biology. The same thing turns out to be true in life. If you want to build a new form of animal life, or if you want to build a little miniature machine, this is that U-joint that you saw a minute ago in the bacterial flagellar motor, you've got to build it out of proteins, and proteins are made of smaller subunits called amino acids, these little knobby things there with the letters on them. And the trick in biology is to get the amino acids sequenced in just the right way. Last night at Biola, I had a really dorky little visual aid that I use a lot, snap lock beads. And since it's an older crowd tonight, I didn't think I could get away with that again. But anyway, the snap, there are all these beads with different colors and different shapes, and it's kind of like that with amino acids. They have all these different shapes and different forces that they exert on each other. And if you get the sequence just right, the protein chain will fold up to form these three-dimensional parts of, for example, a flagellar motor, a rotor, a hook, a U-joint, a drive shaft, etc. And it's that way in all of life. If you want to build new forms of life, you need new proteins. If you want to have new proteins, you've got to have code. You've got to have instructions. So the question is, where does that code reside? Where do those instructions reside? And of course, this question was resolved in the 1950s and 60s by scientists such as Watson and Crick, who first elucidated the structure of the DNA molecule and then discovered, 4 years later, in fact, and this was Crick's great insight, that the DNA contains information in an essentially digital or alphabetic form. Crick's famous sequence hypothesis was put forward in 1957. Interestingly, he was a code breaker in World War II. And he cracked, really, I think, the ultimate code the code of life, and his work was critical to cracking the genetic code. But his 57 hypothesis was called the sequence hypothesis. And what he realized is that inside the twisting double helix of the DNA molecule, there are 4 chemical subunits that are alternately sequenced that are functioning like alphabetic characters in a written language or digital characters in a section of computer code, like the zeros and ones we use today in computer code. Which is to say, it's not the chemical properties of these subunits that matter, or their shapes, or their molecular weight. It's their arrangement in accord with an independent symbol convention, which was later discovered and is now known as the genetic code. This is a robust information storage system that is running the show inside the cell. Now,
in 1957, Francis...
I'd really like to show you this, but maybe I'll give you an advertisement instead and maybe do it descriptively, because it's about 3 minutes, and we've got some even cooler animations at the end of the program tonight. But and plus my narration voice is not the best. But basically what's going on inside the cell is that the information in DNA is directing the construction of proteins, and proteins are the key tools in the cell. They perform all the important jobs. They catalyze reactions at rates much faster than would otherwise occur. They build the structural parts of miniature machines like those flagellar motors that we saw. They also are important in processing and replicating the information in the DNA. So you need DNA to build proteins, but the proteins are necessary to process the information on the DNA strand. It's a classic chicken and egg. Fascinating stuff. Here's an analogy that might help, because I don't have time to show this little animation, but go on my website if you want to see exactly how this happens. In Seattle...
In 1950...
Shut up, Meyer. In Seattle, we have 2 great famous companies. They both use code. One is the Microsoft company. They write code. I see Walter Myers here tonight. He's a Microsoft guy. But then there's also the Boeing company, and they use code to direct the construction of their airplanes. That uses a technology called computer-assisted design and manufacture, where digital code is used to direct the construction of a mechanical system. So an engineer might write some code at a console, will go down a wire, and it's translated into another machine language that can be read at a manufacturing apparatus. And if you're up at Boeing, that manufacturing apparatus might be helping you place the rivets on the airplane wing at just the right place, so you've built things properly. So you've got information directing construction. And that's the process that's going on inside the cell. We've got this digital code directing the construction of the proteins and protein machines that keep all cells alive. Now that raises a really profound question. And I call it the DNA enigma. But the DNA enigma is not, what is the structure of the DNA molecule? We know that thanks to Watson and Crick. It's not where the information lies inside the cell. It's in the DNA molecule, or at least a part of it is. It's not what does that information do. We know that. It's been mapped out beautifully during this period of time called the molecular biological revolution. What is the information enigma? Where did it come from? What was the origin of the information? Richard Dawkins has noted, he of apparent design only, no real design, but he says, the machine code of the genes is uncannily computer-like. Our local hero in Seattle, Bill Gates, says DNA is like a software program, only much more complex than any we've ever created. That's the big mystery in biology. You want to build something, you got to account for the origin of the code, because you can't build anything without the instructions that are involved in this essentially CAD/CAM process. We take biology. We learn this stuff even in high school, but somehow the significance of this didn't dawn on me till much later. This is a stop-press moment in the history of biology. The foundation of life is not matter and energy alone, but information. So, back to neo-Darwinism. This ends up presenting a huge problem for both forms of evolutionary theory. The chemical evolutionary theory that attempts to explain the origin of the first life, and then Darwin's form, the biological evolutionary theory that attempts to account for new forms of life from simpler pre-existing forms. Let's just take the Darwin part. That's supposedly the easier part, getting new life from old life. According to neo-Darwinism, new information arises as the result of random copying errors or other changes in the sequence of those A's, C's, G's, and T's along the spine of the DNA molecule. But how plausible is that in light of our understanding that the DNA is like a machine code, or it's like computer software? What do developers know about introducing random changes to the code they've written. Do you want new zeros and ones at any old place along in your code? I'm getting smiles from the people in the audience that I know are developers. What happens when that? You get bugs and glitches. And if you keep going with that process, you destroy the function of the program altogether. Long before you ever get to a new operating system or algorithm or new program, okay? The same thing turns out to be true for very precise mathematical reasons that we can demonstrate, and I've written this up in my books, in the case of the DNA and proteins. If you want to build a strand of DNA that has information on it for building a new protein from an old strand of DNA that used to build a different protein, you're going to degrade that code long before you get to new functional information that will build something fundamentally new. And if you want to talk to somebody who's done the experimental bench research on this, Doug Axe will be on the podium in a little bit. He's done that. One of the reasons that this is true is that there are a lot more, with computer code and human languages and also the information in DNA, if you have an informational system that's essentially alphabetic or typographic, there are a lot more ways to arrange those characters that give you a functional sequence than there are ways to arrange the characters that give you gibberish. And the ratios are astronomically in favor of the gibberish. That's why when you play Scrabble and you rearrange things, you're not going to get triple word score. That's why when you start rearranging the zeros and ones, you're going to introduce bugs and glitches. The same thing turns out to be true in the biological case, and Doug has actually quantified this with a very formidable and rigorous program of research that he conducted at Cambridge for about a dozen years. He wanted to know how rare the functional sequences of genetic information were in comparison to the gibberish sequences. And to make a long story short, he got a scary number. For every one that's functional, there are 10 to the 77th that aren't for a relatively modest length protein, which means you're overwhelmingly more likely to fail than to succeed in finding new genetic information through the Darwinian process. A lot more to say about that, but I hope we've triggered something. So neo-Darwinism has not been able to explain the origin of new information, new genetic information. There have been a whole bunch of new evolutionary theories that have been proposed to try to solve this and other related problems, the origin of form being one of them. And many of these were discussed at the Royal Society Conference. Many of these were pronounced less than momentous. Let me illustrate with just one, which I think is the most exciting of these new evolutionary ideas. It's an idea called natural genetic engineering. It's the idea and the recognition that many of the mutations that we observe in actual living organisms are not random at all. They're not Darwinian in the sense of being random. They're actually occurring in response to various environmental stressors and a pre-programmed adaptive capacity built into the organism itself. This is the idea of the really prominent and famous cell biologist at the University of Chicago, James Shapiro. He was one of the conveners of the Royal Society event. And he says cells have this ability to react to environmental stress because they've got these pre-programmed adaptive responses that are under algorithmic control. All very cool, very non-Darwinian, an advance in our biological understanding. Agreed, check, check, check. But what question does Shapiro fail to address? Where'd the pre-programming come from? And of course, that's what interests us in the work on intelligent design. Because what we know from experience is that information always comes from conscious activity, from intelligent agency. This is a quotation on page 16 of a small technical volume on the application of information theory to molecular biology. I came across this in my PhD work, and it was, for me, one of those penny-dropping moments. Aha. Because I'd been thinking hard about whether or not the theory of intelligent design could be formulated as a rigorous scientific theory, as a rigorous scientific hypothesis. And I'd learned from my reading of Charles Darwin that when you're trying to reconstruct an event in the remote past, you use a different method of scientific reasoning. It's called the method of inference to the best explanation, where the best explanation invokes a cause which is known to produce the effect in question. And I asked myself a question after thinking about that. What is the cause that's known to produce digital information? That's when I came across this quote from Henry Quasler. He said, nonchalant, offhand, the creation of new information is habitually associated with conscious activity. It's what we know from experience. So when Bill Gates says the DNA is like a computer program, well, what do we know about programs? They come from programmers, right? That's part of our knowledge base. It's part of our knowledge of cause and effect. And that was Darwin's principle of reasoning. When we're reconstructing the past, we should do so consulting our knowledge of cause and effect. The best explanation is going to be the one that evokes a cause which is uniquely known to produce the effect we're trying to explain. What's the effect we're trying to explain? It's information. Whenever we see information and we trace it back to its source, whether it's in a computer program or a hieroglyphic inscription or a paragraph in a book, or even the computer programs that are used to simulate evolution in action, so-called genetic algorithms, the information for that code was written by a programmer. And that's the reason that it works. It's not because you really have random processes that you're simulating. So information is always a mind product. And that's the argument that we've developed that's, I think, the even deeper argument that we've developed for intelligent design. Now, the response to that argument has been really interesting, and I finally got a serious review of my book in Science in the fall after it had been released. This was Darwin's Doubt. It was a respectful review. That wasn't the part that excited me. It was the negative part. It was the criticism. It came from a very serious, high-ranking paleontologist and evolutionary biologist at the University of California, Berkeley. His name is Charles Marshall. He correctly identified my argument. He said, Meyer's case depends upon the claim that the origin of new animal form, animal body plans, depends upon a vast amount of new genetic information. Yep, that's what I'm arguing, I said. He got me, finally. But then he says, that's not our present understanding. Sorry for the typo here. He said, our present understanding of body plan building, or morphogenesis, is that the new animals were not made by new genes, but by the rewiring of gene regulatory networks that act on other already existing genes. A gene regulatory network. I didn't get to talk about them, but gene regulatory networks are networks of genes, each of which are full of genetic information that are essentially functionally integrated information processing systems that control when other genes are turned on and off. The other genes also full of genetic information. Anyone see a problem here? In order to explain the origin of new information without an intelligent agent, Professor Marshall just invoked other sources of pre-existing unexplained information. Now, when people do that, it's called begging the question, and you don't need a PhD in biology to identify the fallacy there. And so we think that, whether it's Shapiro invoking pre-programmed adaptive capacity or Marshall invoking gene regulatory networks that already exist, which are not explained, we think the materialists are in serious trouble trying to explain the origin of the information necessary to build new forms of life. So we think we have a powerful argument here. Now, we get to the new stuff. We call it ID 3.0. ID 1.0 was Phil Johnson and Michael Denton, who wrote some of the original books, and Charles Thaxton, my mentor, who wrote some of the original books advancing the idea of intelligent design and critiquing Darwinism in a rigorous intellectual way. ID 2.0 started at the Discovery Institute when we began to fund books like Michael Behe's Darwin's Black Box or Bill Dembski's The Design Inference, books that laid out our core case, our argument for intelligent design. My book signature in the cell was kind of a lagging, a trailing contribution to that effort. But we had a core of really good books that have made the case for intelligent design. We've made the argument. But when our opponents can do no better than beg the question, we don't think we need to, what is the flog the dead horse? No, I don't think I got that metaphor quite right. Anyway, with a flagellum. Thank you, Howard. Yeah. Beat a dead horse. We don't need to keep making the same arguments over and over again. Doug has also expressed this impatience. Why do we keep needing arguing? We won the argument. It's obvious to anyone who's fair-minded. Let's now move on. We'd like to use these core concepts of functional integration or irreducible complexity, or the idea of functional information, or what we call technically specified complexity. Let's use those ideas to start to make some discoveries. If we think life is designed, it ought to look different when we go and investigate it carefully than if it arose through a series of gradual, undirected steps or processes. One of the first predictions that we made in the intelligent design movement of that sort concerns something called junk DNA. Anyone heard of it? Okay. got a lot of press right up to, I was writing Signature in the Cell, 2009, people were still talking about junk DNA and how it proved that Darwinism was correct because there were these big sections of the genome that didn't seem to code for proteins. And therefore, they must be non-functional, and therefore, that's the expected byproduct of that trial and error process that we neo-Darwinians had expected. Random mutations accumulating over time. Well, we thought, well, okay, we believe that mutation is a real process, and we believe that natural selection is a real process. But if the genome was designed, we wouldn't expect to see 97% of it not coding for being junk and only 3% being functional. We thought that the signal should dwarf the noise, not the other way around. And so we predicted that the non-coding regions of the DNA would turn out to be importantly functional. Richard Sternberg was at the forefront of the work on junk DNA. He was the guy that ended up leaving the Smithsonian under pressure, let's just say. Anyway, in 2011, a major study came out of the National Institutes of Health called the ENCODE Project. And it confirmed what Sternberg and a co-author, the same James Shapiro I mentioned a bit ago, had been writing for years. Sternberg predicting it on the basis of ID, that the non-coding regions are going to have a function. And it was a massive study showing that at least 85% of the genome is transcribed, probably more, probably north of that eventually, and that it's transcribed into various RNAs that have lots of important regulatory functions because it's not just the DNA, it's a whole complex, hierarchically organized informational processing system. Professor Shapiro graciously acknowledged Richard Sternberg as the first one to have the insight that the non-coding regions would be functional. He says, though we have different evolutionary philosophies, I need to acknowledge that my colleague, Dr. Sternberg, saw this first. He saw it first because he holds to the view that life is designed by an intelligent agent. Now, let me give you some other examples of things that we're now doing. Doug and I have a colleague who's a brilliant Microsoft architect. His name is Brendan Dixon. He's a tall guy, 6'5", wild shock of Einstein hair. And when you talk to him, you think he's probably about that level. And anyway, he was writing code for Doug to simulate the gene expression system with a computer model. And he wrote 10,000 lines of code for us. And one day he walked into my office and he threw a book down on the table called Design Patterns. And the software developers here will know what that is. I didn't at the time. And he said very dramatically, I get an eerie feeling that someone has figured this out before us. And by this, he meant all of the design patterns that he was discovering in the function of the information and the way it was processed inside the cell, which he was now modeling with his code. And he then explained to me that the book Design Patterns was a standard operating or design manual for computer developers, for software developers, and that a design pattern was an established method of storing or processing digital information. For example, he said, and then he ticked a bunch of them off, but just for your benefit, one such design pattern is the automated error correction. You know how you have a spell check that you can run, okay? Well, the cell has all kinds of proteins, as a good doctor, a protein chemist was explaining to me before the talk started. 200-some proteins that are involved in making sure that the DNA is copied properly. And if it isn't, if something gets inserted that's not supposed to be there, there are proteins that come in, pull it out, and put the right nucleotide in there. So we've got automated error correction. We have hierarchical filing of information, said Brendan Dixon. We have nested coding of information, an advanced way of encrypting one message inside another. And he said, I get an eerie feeling that someone figured this out. And then he went into Microsoft talk, and he said, only it's like, it's the same basic design patterns, only they're being executed with an elegance that transcends anything we've achieved. It's like an 8.0, 9.0, 10.0 version of our own advanced design principles in computer science. So one of the things we've started doing to take the design concept and run with it was we started looking for design patterns. Maybe some other ones are out there that we haven't found yet. Because there's lots of design patterns you can, we know what software designers do to process and store and use information. So maybe there's some other ones out there. And there's some scientists, and unfortunately, with all the other projects I'll talk about tonight, I will show you the scientists, but with this one I can't. I will just tell you, the scientist who's heading this up is a very prominent scientist, not in America, he's in Europe. He's headed a very large lab. He's working collaboratively with 2 other major groups in Europe. And basically, he's looking for what George Washington used to do. Remember, well, this is a little book we read to our kids called George Washington Spy Master. If you send a message, he'd write out a nice, innocuous message about, remember to, Martha, hang up the laundry on Tuesday. But if you'd start reading that same message 1 letter over, what biologists would call a change your reading reference frame, then there was another message. And that was the real message. So if you knew the cipher, if you knew the rule for how to read the message, you'd find the real message embedded in the message. And what this scientist began to look for, he thought, well, maybe there's some, if the cell was designed, and if we're looking for efficiency, then an efficient designer might build messages on top of messages so that you get a really dense information storage. So he started looking for overlapping genes in bacterial chromosomes. And he found that in every reading reference frame he looked, there are actually 6 possibilities, 3 going forward and 3 going back. He found multiple messages, genetic messages, that coded for other proteins. So this is just a stunning discovery. He had an atheistic postdoc in his lab who was asked by people in an adjacent group, well, what do you make of this? They were actually having trouble getting the results published at first because people realized it's hard enough to explain the origin of the information in just 1 reading reference frame. But if you've got messages layered on messages, you know how hard that is? I mean, by chance, you're going to get, because you start, if you don't get one right, if you try to change it and then write another message, you're going to wreck the other message. So you got to really think that through, how you're going to layer it and make sure all of them are functional. In any case, this atheistic postdoc was asked by other researchers, what do you make of that? And he just went, I'm rethinking my worldview. Yeah. Anyway, that's one of our projects, and I've got some pictures of it, but it's a really exciting project. There's been some publications coming out, but we're actively supporting this financially behind the scenes, so I have to be kind of quiet about this because it will not help the scientists to have our support for him known. Here's another part of the information idea as a guide, what historians call a positive heuristic, a guide to discovery. According to the Darwinism, every section of DNA that's functional should have evolved from some other very similar gene and from, in turn, some other similar gene by a series of mutations. So you'd have lots of different mutational processes that would produce one gene, would gradually change one gene to another. Now, if you think that the genome was designed, however, you wouldn't think or require that every section of functional DNA would have evolved from something else. It might have appeared abruptly, or it might be discontinuous in its relationship to other information. When we write a book, we don't gradually change all the letters from some other book. We don't see a series of letters changing from Hamlet to Stephen Hawking, for example. Hawking wrote his own book and started basically from scratch and put things. So there's no homology, no similarity there's maybe a little bit you could find, but there's going to be a lot of unique sentences in Hawking that you don't find in Shakespeare, right? So a design approach would expect to find discontinuity in these sequence comparisons, whereas a Darwinian approach would expect to find continuity. So we thought, well, let's see. Let's go and look. Are there what are now called orphan genes out there? Technical people will know that orphan stands for open reading frames, but it was a play on words in biology. It meant genes that don't have any parents, genes that aren't related to other genes. So it started coming out in the literature in accord with things that we actually expected, that there were more of these orphan genes than you would expect. They were turning up in lots of different types of organisms. So here's a Darwinian expectation. If you got a functional protein, it should have some similarity to other known functional proteins. But what we're finding instead is the expectation of design that there be a lot of things that have no relationship, genes that have no relationship to other genes. So we've initiated a project on this, and it's turning out that these orphan genes are ubiquitously distributed across the phylogenetic hierarchy. They're turning up everywhere. I took the slides out in interest of time, but one really interesting, cool example came out in a scientific paper. You've got these two mollusks that are really closely related. They have proteins, about 100 proteins that are involved in building the shells of these mollusks. They look similar. They're very closely related subspecies, and they have only 19% of the same genes and proteins. The other 20 or 80+% are orphans. Deep puzzles for evolutionary biology, but things that are really impressive from a design point of view, because we know designers can write new code. And we can, I'll do it this way, I'll write a manual this way, So anyway, one of our big projects, we're automating a search right now. We have 4 key researchers, scientific researchers, and a whole team of programmers working on this, including some wizards in Sri Lanka that are just awesome. But our core scientists are Richard Gunasekera from the University of Houston, Change Tan from the University of Missouri, Paul Nelson, who's been with us for a long time, and Richard Buggs, recently tenured professor at the Queen Mary College at the University of London. He had a piece on orphan genes in the trees he studies, ash trees, and then wrote a blog on the Nature. It was published in Nature, and then he was allowed to write a blog on the Nature site explaining why these orphan genes were a challenge to Darwinian theory. So we're starting to break into the mainstream on some of these projects. Okay, that's some of the stuff on the information. And in the time I have left, let me give you some stuff that's just really exciting. It's using the irreducible complexity, looking at molecular machines. Here we go. Let's call it using rational design principles to understand the function and to redesign nanomachines. We're going to understand the design better using rational design and engineering principles, and then we're going to use some of those principles to copy the kind of design principles we see in nature and then use that to solve some problems of our own. That's a field of science that's known as biomimetics. It's using what's in nature to solve design problems that we face. Because after all, what's the premise? Nature was designed, life was designed. So let's appropriate some of those design principles ourselves. So I'm going to have Doug come up now for the last part, and I'd like to show a little animation of a machine that is as exquisite and interesting in its own right as the bacterial flagellar motor with which we started the evening. This is called an ATP synthase. It's an energy-generating turbine, and it builds the ATP molecules that store the energy that drive all the important biochemical and metabolic reactions that all of us need to stay alive. Doug, I thought I'd show the animation, then I'll let you, as a trained Caltech chemical engineer, comment a little bit on what he sees and understands about this. Just by way of introduction, Doug's, when we have external projects, we have some of our core scientists assigned to those projects to work collaboratively with the external researchers. There's a biophysicist in the Department of Physics at Cardiff University, Amir MacDonald, who approached us not very long ago and said, hey, I'd like to get involved with the ID research. And this has been one of the really neat things for us, is that we spent these years writing these books, and now we're finding senior professors saying, hey, I'm kind of intrigued with your perspective. I'd like to push it forward in my research. And that's what our ID 3.0 is about, these projects we're planting in major universities. But MacDonald is maybe a little younger in that picture, but he's 55, senior professor, tenured, and he's working on this ATP synthase. And so we're going to show it to you and then let Doug explain what an engineer sees.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology. A rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible. ATP synthase. An example of intelligent design.
Yeah, so what do you think?
Isn't it?
Yeah.
Am I on here? Yes, I
Yeah,
am.
you're on.
A couple of things as I'm watching it, and I haven't watched that for a while, so I was watching with you. In order for you to even watch that, it has to be slowed down by a factor of 1 million or so. So it's not going like this. Remember, 100,000 cycles per second. Another thing that's interesting about just visualizing, just making these things so that you can see them, they're actually too small to be seen. And when I say that, I'm not just saying that you'd need to magnify them. I'm saying they're approaching the size of wavelength of light. So they're actually too small physically to be seen the way we see other things. You have to use an electron microscope to get even a crude image of these things, and you have to go beyond that to actually figure out what they look like. Another interesting thing, respect in which they're more complex than what the video shows, is those look like blobs of clay, right? Each piece looks like a blob of clay, whereas each one of those pieces is actually a long chain, hundreds, sometimes 1,000 amino acids of precise sequence in order for this whole chain to form, fold, collapse upon itself, and form a structure that's overall shape is like that, somewhat like that blob of clay, but there's internal structure that's making that structure. So it's far more complicated structurally than that. And if you've done any, if you study biochemistry, one of the things that everyone recognizes in the textbooks say that enzymes do, and these are proteins that do chemistry, is they speed up reactions. But there are, so you might read in a chemistry textbook that an enzyme is something that accelerates chemical reactions, but we make simple catalysts that also accelerate chemical reactions. They don't just accelerate, they accelerate to a phenomenal degree, but this is a classic example of something completely beyond accelerating a reaction, and that is coupling reactions. So in this case, there are two, I don't think, did you talk about the protons going through?
No.
There's two things happening here. Protons are flowing through the membrane because there's a gradient. It's more acidic on the outside than on the inside, and so H+, protons are flowing through. They can't get through the membrane, so they're coming through this motor because it allows a channel for the protons to come through, and the energy that's liberated as these protons flow is being used to drive that drive shaft. And the structural pairing between that drive shaft and what it does to the things that it moves out as it's rotating around is what enables the ADP and the phosphate to bind. So you get this elegant coupling between two things that by all rights have nothing to do with each other. One is protons want to get inside the mitochondrion, and the other is the mitochondria wants to make ATP. Those are totally unrelated as burning gasoline is to taking the family up the mountain to go camping. Those have nothing to do with each other, right? Burning gasoline does not take you up the mountain unless you've been very clever about making a machine that burns gasoline in order to take the family up the mountain. And this is precisely that sort of thing.
That's brilliant, brilliant. So Professor MacDonald at University of Cardiff has worked for much of his career characterizing the structural parts of this system and explicating how it works. There's also a Japanese group that has done the same. And when he approached us, he said, I've come to the conclusion that Michael Behe was right. I think with my system, he says, it's been so extensively characterized, I don't think there's any possibility now. There's no way to argue that this arose through a series of gradual steps. And he's writing now a number of technical papers on the design features of this system and also how we can learn from those design features to maybe perhaps do some of our own design of nanomachinery, which segues to Professor Jim Tour. You've already seen Professor Richard Ganesekera. He's at the University of Houston, an extremely versatile biologist who will, as it happens, be coming to Biola University. And, but Professor Ganesekera has been assisting Tour as well as doing he's one of the leaders on the Orphan Gene project, but he's also working on this project in nanoengineering, nanotechnology. I'm going to talk about some other nanomachines. Tour, a few years ago, started writing articles expressing skepticism about evolution at a chemical level. And he kind of did the kind of dumb like a fox Southern lawyer thing and said, you know, I'm just a chemist, I'm not a biologist, but I don't see how this works at a chemical level. Somebody help me understand this. And a young guy who worked for the National Center for Science Education named Nick Matsky sort of took up the challenge, and it was kind of foolish on his part. Tour is one of the top 3 or 4 organic chemists. He's an organic synthesis guy. He's got 600 peer-reviewed publications, 150 patents. Matsky was working for this advocacy group. But curiously, we don't know exactly how or why, but Tour's nomination to the National Academy was soon held up. Later, he lost some funding that seemed to be a shoo-in. And he got David Berlinski heard about it, got in touch with Tour. Little by little, we got to know Tour, and Tour started writing some really trenchant critiques of the theory of chemical evolution, saying, I don't see how this works from a chemical point of view. How do you get the chemicals in the prebiotic soup to arrange themselves into DNA and proteins and cells? Because chemistry doesn't want to do that. Chemistry goes in the opposite direction. You have to constrain chemical reactions to move in a life-friendly direction. Anyway, at the same time, he was working on some little and he knew this because he was making his own little miniature machines. Not machines that were flagellar motors or ATP synthases. They were little nano cars, using things of the size of carbon atoms as wheels and axles, and they actually have nano car races now among nanotechnology specialists. This is a form of bioengineering. So this was kind of a play. I mean, it was a really interesting technology. It's very hard to get the chemical reactions to happen to build specific structures, and that's where he became skeptical about chemical evolution. But then he got to thinking, well, what if I disassemble that nano car and use the wheel and the axle more as like a drill or an auger? And he started, thinking of some rational design approaches to some medical problems like cancer and infectious disease. And what Dr. Tour has done, with some help from Richard Ganesekera, who's a specialist in the bacterial side, is he's made now not a nano car, but a nano auger, a drill that will drill through the cell wall of a cancerous cell, lyse it, and destroy it. We can see the contents oozing out when they do this. Now, this was a very difficult thing technologically because you got to get the little drill to bind to the cancer cell, and that's an organic chemistry thing that requires some expertise, but that's what Tour does. So he figured out how to get them to bind to the cancer cells and not the surrounding healthy tissue. And he's also figured out how to attach to that guy at the other end a switch that will activate the spinning, the turning. Activate it by light. So you activate it with different frequencies of light. Now, light has a depth of penetration issue, so the next stage in their research is they're trying to also activate it with certain frequencies of X-rays that are not too intense to damage things, but just right to turn on the switches. So it's a kind of engineering trade-off thing. But he's using principles derived from studying the nanomachines in nature to redesign nanomachines to solve some pretty important human problems. And I have a little animation that Tuur and his folks at his lab have produced. He's at Rice University. And if you follow Science Press last summer, he started to get some amazing press. There were about 10 different articles in the science press around the world about this breakthrough technology that he's developing. He's also a very courageous guy. And he'd taken some hits because of his expressing skepticism. But when we had some long conversations and offered to support him in this research, for which he'd lost money from the National Science Foundation, he went to the president of the university at Rice and said, listen, I know these discovery guys are radioactive, but they're doing good science, I respect them, and I want to accept the grant that they're offering. And I want to know if you have any problem with me doing so. The president of the university went right to the biology department, who had already pitched a fit, had some conversations, did his own research, and came back to Tuur. And maybe not because we're so cool, but maybe because Tuur has got 600 peer-reviewed publications and 150 patents, said, I approve. And so in every publication that Jim Tuur has done with our support, he has acknowledged us specifically as the funding source. So we now have, last year we had a great year. Richard Buggs, whom I mentioned, Gunter Beckley, a paleontologist that I haven't had time to talk about tonight, and Jim Tuur have all had publications in nature-related journals, Tuur's in Nature Nanotechnology, acknowledging the radioactive toxic Discovery Institute. So we're kind of pleased with that. But much more important than...
What's our half-life?
Yeah, what's our half-life? Yeah, thank you. Anyway, much more important than that is what he's doing with all this. And so let me show you this little animation that his group has developed. And I might get Doug up here
Molecular
again
nanomachines
in a minute.
are tiny machines that can drill holes into individual cells and deliver therapeutic agents. They are so small that we could place 50,000 of them across the diameter of a human hair. We increase the functionality of the nanomachines by including add-ins that recognize a specific cell surface and then bind the nanomachines to that surface. For example, we used peptide add-ins to recognize the surface of human prostate cancer cells. As the nanomachines approach the cell's surface, they bind using those peptide add-ins. We can then activate the nanomachine by exposure to light, and the rotor of the nanomachine will spin at 2 to 3 million rotations per second and start to burrow its way into the lipid bilayer, leaving a hole in its wake. Upon entry into the cell, the nanomachine can either deliver specific drugs through the hole to begin to treat the cell, or continue to shine the light and have the nanomachine drill further into the nucleus of the cell to rapidly kill the cell. In the latter case, the cell quickly shows blebbing, where it starts forming large protrusive bubbles on the cell surface and then shrivels and dies by necrosis. In principle, we can target any cancer cell type or any other particular cell through the variable peptide recognition add-ins to cause the cell's death within minutes upon specific light activation. This represents a new molecular mechanical approach to the treatment or death of cells.
Anyone see the potential of that? There are 3 forms of treating cancer today: radiation, chemotherapy, and surgery. This holds the promise of a 4th, and it's pretty exciting. It's pretty exciting. Doug, you want to come up? We can just have a little conversation about this, and then we'll open this up for discussion. These are just, I think, I mentioned the junk DNA as well, but we've just 4 or 5 of our new research projects. We have 15 that we're pursuing. There's a lot more, but this is, Doug and I were just having a conversation about this today, about the different approaches to cancer research. And you were contrasting this with the typical approach of, what do you call the combinatorial approach?
Yeah, combinatorial chemistry has been popular for a long time. And basically, they don't talk about it this way, but the philosophy behind it reminds me of a theory you may have heard of, where you start with just a random gamish and then you throw it at cells and pull out something that works. It's very much a Darwinian sort of idea that instead of trying to figure out what would work, you have chemists produce a mess basically, that has millions of different chemical variants, and then you filter the mess to see if anything in that mess is active, and then you might do some variations on that mess. Tour's approach is very, very different and very much more a design inspired approach, where instead of saying we don't know what to do, so we're going to try everything and see if anything works, you actually build a machine that has the potential to do something here, and then find a way to target the machine to the tumor cells, which I think is...
And one of our key concepts in design is specificity. In fact, one definition of information is called specified complexity. And what organic chemists do is they use their knowledge of the particular chemical properties and three-dimensional configurations of molecules to those add-ins they were talking about, to bind things with specificity. So this is a rational design approach, which is kind of cool. It's also employing a lot of knowledge of the principles of the nanomachinery in order to build new nanomachines. So it's also biomimetic in that sense. There's another aspect of this that where we think intelligent design has a lot to offer in cancer research, and that is exploiting our knowledge. And Doug has done some of this really cutting-edge research. It's called the waiting times problem. And it turns out, I was going to tell more about another scientist, but in interest of time, waiting times is basically this, that there is a mathematical underpinning to neo-Darwinism called population genetics that allows you to calculate the amount of evolutionary change that you ought to be able to, that you could reasonably expect in a given amount of time, or conversely, the amount of time it would take for a given amount of evolutionary change. If you know the mutation rate and the population sizes and generation times and so forth. This Darwinian math that underlies the theory is actually posing a real severe problem to the theory right now, because as we've learned that there are many structures that can't be built unless there are coordinated mutations, the waiting times go up exponentially when you need 2 or more mutations to happen in a coordinated fashion. And so that suggests limits to how much change can occur in a given amount of time. And if we exploit our knowledge of the limits of the ability of the mutation selection mechanism to generate change when it has to produce 2 or more changes, that suggests that maybe there's some therapeutic approaches where we target, say, a bug that's become antibiotic resistant with 2 or more drugs, drug cocktails. We're doing this already with antibiotic resistance, but a similar approach, there's an ID-friendly guy at the National Cancer Institute that we've been talking to that says, no, he says all your stuff on limits of evolutionary change, exploiting the limits of evolutionary change, could be really relevant in cancer research, because we can devise therapeutic cocktails that are going to exceed the ability of the cell to replicate out of its problem and to mutate out of its problem. So there's some potentially very powerful approaches to some really serious medical problems coming out of ID research. Tours is the most exciting right now because they're doing this. You know, they're destroying these cells right now. So anyway, these are just some of the projects that we're working on that show some of the promise of intelligent design. And we thought we'd open it up to an open Q&A, questions that can be directed to Doug or me. And we don't just take questions. We also take comments, objections, abuse. It's all good. Yeah. So, yeah. Thank you very much, by the way. It was lovely to be with you.
Hello. Is it okay? So commonly you hear that the difference between monkeys and humans is something like we're 99% the same genetics. I'd like to hear your comment on that type of.
Yeah, and I may pull some software designers in on this. We were talking about this at lunch today. That number is going down. First of all, 95%, maybe lower, as you take more account of the non-coding regions. If you exclude those and assume that they're non-functional, the numbers are higher. But I've been talking to developers about this. And in fact, it was at our meeting last year, right after the Royal Society, where we had those British software developers. And they were saying, we're not impressed by that argument at all. Because we know that when we write code, a big portion of that code is common no matter what program we're running. Because you just need a big chunk of it to run the basic machinery, the hardware, right? Your operating system. All that stuff is common. And the part that differentiates one program from another is usually a very small percentage of the total code. So Ann Gager gave a fantastic talk last night at Biola about all the differences between chimps and humans. starting with art and science and football. And you know, moving, and I mean, the cognitive abilities, our ability to use language, you can't account for those differences on the basis of if our genes are the only thing that make us different, then 5% doesn't get you even close to counting for the anatomical, behavioral, and cognitive differences. So something else has got to be going on, and it turns out that the way the genome is regulated, the epigenetic or ontogenetic, the higher level information, there's a lot of differences. Our proteins are very much more different than our genes, which means they're being processed differently. This is the work of our colleague in Brazil, Marcos Eberlin. Doug, any comments? And I would love to get Walter's comments on this too, as a software guy.
Well, I mean, most of the code you're writing today, you're using base libraries. So anywhere from probably 60 to 90% of the code that you're writing, there already existing libraries that you use when you're writing your code on top of that. Don't start from scratch.
Yeah. Yeah. So it'd be poor design if you had 90% different. If you're, yeah.
Yeah, so I mean, you need common functionality across them. So in terms of a human or an ape, there's a lot, there would have to be a lot of commonality in terms of what they do. But they're quantitatively different things as well between those two that have to be a result of some additional type of code that's written on top of the base code.
So the difference that makes a difference is a small percentage.
It can be 10, it can be 40%. It really depends on specifically what you're writing. But since there's so much, I say there's a lot of common libraries. I would just say there's a lot of common libraries, if you will, between the humans and apes. But still, that part that's not the same, that had to be coded. That was completely new code.
Doug?
There's also a presumption, typically, when we talk about this, that our genome is a blueprint for making us. And that's actually not a proven fact in biology. That is an assumption. And it's an assumption that I question because I don't think in 4 billion bases, which would be 8 billion bits of information, you actually have enough information to specify a human being. If you consider, for example, that there are a quadrillion neural connections in the human brain, that's vastly more neural connections in the human brain than there are bits in the human genome. So even if the genomes are quite similar between, say, us and chimpanzee, that doesn't...
There's got to be other information.
There's got to be something else going on that makes us what we are and what makes Pan what Pan is. Chimp.
That's very good. Very good. Yes. Wayne.
Just for clarification with James Tour, is he openly declaring that the research he's doing is based on an intelligent design framework? Or is he, I mean, because if he cures cancer...
No, I would say it's implicitly design friendly. He's an evolution skeptic and pretty much a nuts and bolts science guy, so he's not wanting to wade into the big, debate with the larger theological and philosophical implications. But I think that's important to state. He said, well, I'm really not taking a position on ID, but he's using design principles, which is why we're supporting him. He also, I think he's very friendly to what we're doing, but it's kind of like, I want to do the science, I don't want to get into that debate, but he's modeling the kind of science that we think is what flows from an ID perspective.
Gotcha. I was thinking Jim Shapiro when you said James Tour, different.
Yeah.
Jim Shapiro, he's the genetic toolbox guy, is not at all wanting to be on our camp. I'd say Jim Tour is
increasingly sympathetic.
Well, when it comes to the origin of life, he's just saying there's no way you can do this unless you know
He's actually got a new article coming out in David Berlinski's journal Inference, calling for an end to prebiotic simulation experiments and origin of life research. He thinks it's a complete dead end. And that seems anti-intellectual, except in the history of science, there are many cases where nature has spoken definitively and we decide, okay, we're not going to do that kind of research anymore. We don't fund research on perpetual motion machines. We believe in the conservation of energy. And Tour is, in fact, saying what Dembski's been saying. We believe in the conservation of information. You don't get it unless an intelligence puts it in. So let's just stop this nonsense and move on to not waste money on this when we could be doing something more productive, like trying to figure out how to stop cancer. Amen. Yeah.
Thank you.
I have a question about how information is expressed in social insects. For example, if you put a leafcutter ant on the ground, it'll wander around aimlessly until it starves to death. But if you put 10,000 of them together, they can suddenly develop problem-solving abilities such as building bridges and ladders up to your food supply and so forth. So what is it that makes the information, which is apparently inherent in each individual, inexpressible individually, but expressible as a group?
That's a tremendously astute question, and I do not know the answer. That sounds like a research project, but I actually can turn you on to a scientist who does. His name is Scott Turner. He's written a new book called Purpose and Desire, which is about the concept of homeostasis. He's an not paleontologist, an insect biologist, and he studied the social behavior of termites and ants, and so I'd get his book on this. It's really quite good, but I don't think I can answer that question myself. Doug?
I can't either, but there's something called swarm intelligence. I don't know if you've come across that. So there are people are playing with this in computers, that you can have a behavior that's fairly unremarkable as a single thing, but if you get a large number of things that have the same sort of instinct, and you put them all together, what comes out of that can be much more interesting than what a single organism does.
A speculation is that it might be something like a pre-programmed adaptive response where the behavior of the other insects is triggering a response, which in turn triggers a response, which in turn triggers a response. Pretty soon you get a group dynamic. But clearly these kinds of behaviors they're innate. They're pre-programmed, and that's a big take-home in Scott Turner's book. But I recommend it. That's a really well-put question, but it might be a good research program. Yeah. Don't know. Yeah. Oh, yes.
If I may just continue on this subject, I think
the protein biochemist I was telling you about.
If we recognize that there are two ways we can learn from personal experience and from the experience of another, that means that every level a living organism has these two ways of learning, regardless of how complex the organism is. That also implies that the organism must be able to recognize the other from whom the organism is going to learn.
Yep.
That also implies that at the foundation of life is trust, because you have to be willing and able to trust one another in order to be able to learn. If, therefore, at the foundation of life is trust, how do we then impose this idea of struggle for survival?
Excellent question.
They're opposite philosophies, aren't they? That one is that everything is inherently incompatible. I agree. And if you look at, there's an interesting paper that I co-authored with a Finnish biotechnologist, enzyme expert named Matti Laisala, that was on lignin, which is, after cellulose, the most abundant organic, non-fossil organic compound on the planet, because all the woody plants, the stem plants, have a ton of lignin and a ton of cellulose in them. And he spent his career looking at how to break down lignin in order for the paper industry, lots of birch trees growing in Finland. And they found it to be very, very difficult because lignin is such a complex structure that it's very hard to get something that will break it down without ruining the thing you're trying to get to by breaking it down. And he posed the question, there's so much energy on planet Earth that's tied up in lignin. Why is there no organism, so far as we know, that has evolved the ability to use that energy? There's white rot fungi that break down lignin, but they don't get energy for it. They have to expend energy to break it down to get to the cellulose. There is no known organism that can live off of lignin, despite the fact that it's a huge fraction of the total organic energy on the planet. And I thought, that sounds very interesting. There's something that would be exploited if this is a dog-eat-dog world, and if anything can happen by evolution, if anything can be invented, something would have invented the ability to eat that and get calories from it, but nothing has. And he said it's also interesting that lignin, because it breaks down so slowly, that's what enables us to have the layer the humus layer in soil that allows air, water, soil mixture that allows all of the stemmy plants to survive. So if there were something that could rapidly break down lignin, we would lose the topsoil layer, you'd lose all plant life, and you'd very quickly
The whole food,
lose
the whole food chain.
Yeah, everything would go extinct. But who cares? If it's dog eat dog, right? Something should start eating it. I don't care if everything... it's interesting that it doesn't work that way, that things are in balance as though the balance is almost designed.
To favor us. Clay.
Curious, one of the things, if we go outside and pick up a rock somewhere out in the mud, you're going to find life under it. Have scientists ever been able to, you mentioned origin of life research, have scientists ever been able to create anything alive without using already existing organic matter?
No, not even close. Not even close.
You know, you might, what's his name? Venter.
Craig Venter, yeah.
Craig Venter and coworkers claimed to have invented a new life form, but what they really did was they took an existing life form, modified a chromosome, stuck it in. So that's not, they didn't create life, they took life and modified it.
The other thing that's worth commenting on in these origin of life simulation experiments is that even to build molecules that are life-tropic, moving in the right direction of life, a huge amount of intelligent intervention has to go into the experiments. And so what's really being simulated is, I think, the need for intelligent design. There's one of the new theories of the origin of life is called the RNA world, the idea that the first self-replicating system wasn't a whole cell, it was just a molecule of RNA that could copy itself. But in the laboratory we haven't been able to get RNA, we haven't been able to design RNA molecules that can copy themselves, but some scientists have succeeded in getting an RNA molecule that can copy about 10% of itself. But to do that, they have to sequence the bases, the nucleotide bases themselves. So they have to provide the information to get even that limited replication ability, the 10%. So what's being simulated? I think intelligent design. So it's another data point that's showing that connection between intelligence and the generation of information that we say that justifies our design inference. Way back there, yeah.
Yeah, just a question
That's
about...
Jack, isn't it?
Yeah.
Yeah, good.
Just a question about dissemination of information. Like, I'm thinking high schools and colleges. I mean, you have to be thinking about how to get this information out. And just saying, I'm a guy who teaches in South LA, high school and college, and just the thought, rather than starting at Harvard or someplace where there's existing prejudice and, like you say, you're toxic. But there's a lot of schools in my neighborhood that would love textbooks and teachers and resources and animation, and they probably don't have any stigma towards you at all, you know, and I just don't know if there's anything developing with that, and not just a suggestion. You got people, I think, that'll be ready for anything that will be given to them because there's lacking resources in urban schools. And
let's talk after. We have a lot of resources both for teachers in a public school context and in a private school context. One book that we've done called Explore Evolution teaches the arguments for neo-Darwinism as you find them in the current literature, and the scientific criticisms as you find those criticisms in the literature. Doesn't get into the intelligent design question, just the pro and con about the standard theory, and it's been a very effective textbook for use in the public schools. It avoids legal challenge and misunderstandings of church-state relationships and all that, but it also blunts the Darwinian indoctrination. It's pretty good science, really nicely illustrated book. So, yeah, and we got a lot more where that came from. So, yeah, let's talk. Very good point. Thanks for what you're doing in the schools. Yeah, over here. Yes, thank you.
So when you were talking, you talked about like two different kinds of like ways of evolution. You said there was like novel information and then just small changes. What would be the actual difference between those two?
Novel changes versus small changes. Oh, the micro versus macro evolution. Yeah, microevolutionary processes that we can observe, like the finch beaks or speciation, are occurring as populations are isolated from one another by mechanisms, one called geographical isolation. And so you have a parent population that has a large component of genetic information in the gene pool. The populations are separated, and the daughter populations have some but not all of that original population, and so, or that original complement of genetic information. So there's a fixation of different traits, but and so it looks like evolution in the sense that things have changed from the beginning, but it's occurring as the store of information is diminishing from the parent population to the two daughter populations. So microevolutionary changes are occurring as the result of a loss of information, whereas the major innovations in the history of life, where you get fundamentally new things like arthropods in the Cambrian, or the first birds in the avian revolution, or the first mammals in the mammalian revolution, that's requiring a huge infusion of new information, both genetic, and as Doug was indicating, there's more and more evidence that the information in DNA alone is not sufficient to build a whole new body plan. So also we call that epigenetic or ontogenetic information. So microevolutionary processes are taking place because of a loss of a pre-existing store of information, where these macroevolutionary or macro innovations are taking place, are requiring an input of information, and that's what the neo-Darwinian mechanism doesn't explain. That's a very good question. Thank you. Yeah, to the right.
Thank you. I was wondering, among people who are willing to open-mindedly listen to the arguments on IND, what is the most common objection that you hear from people that listen to everything thoroughly, and they look at it and they say, okay, that makes a lot of sense, but what is the most common argument?
I'll tell mine, you tell yours. How about that? Okay. What we most often hear is pseudoscience, or it isn't science, but that's not science, or that's religion, an attempt to label the argument out of existence. And there's a couple problems with that. One is to say something isn't science requires having a definition of science. The scientific method has been famously difficult to characterize definitively or normatively, because it turns out there are many different scientific methods. The method that we're using is the very same method that Darwin used in The Origin of Species. That's what's a little bit mischievous about my two books, is that I'm using Darwin's method to establish intelligent design. It's the method of inference to the best explanation, sometimes called the method of multiple competing hypotheses. So one response I have to that objection is to say, well, if my method of reasoning isn't scientific, and the theory derived from it, intelligent design, isn't scientific, then you'd have to say the same thing about Darwin, because he's using the same method of reasoning. But the deeper objection to that objection, the deeper response, is that the fundamental obligation of the scientist is not to limit himself or herself to a strictly materialistic set of explanations. And that's always the claim. Well, you're invoking a non-materialistic cause. That's not science. Or you're invoking an unobservable cause. That's not science. And there's a lot of unobservables in science, from quarks to fields to unobservable transitional intermediates in the distant past in Darwin's theory. But in any case, the claim is no materialist, it's got to be strictly materialistic or it's not science. Well, we think the fundamental obligation of the scientist is not to come up with the best materialistic explanation. It's to come up with the best explanation, period, the one that's most likely to be true. So we want to say, get rid of that silly rule. Let's follow the evidence wherever it leads to the best explanation. If it's a materialistic process, so be it. But if it requires invoking intelligence to explain the phenomenon by reference to what we know about cause and effect, then let's be open to that. We want a truth-tropic, truth-centric method, not one that is biased towards a particular worldview. Doug?
I would only add that you asked about people who carefully read everything and are willing to consider everything. I think with very few exceptions, people who do that are not antagonistic toward intelligent design. They may have some questions, but they're very open to dialogue after that, and they tend to really favor that approach. The other people tend not to go that far. They don't want to read it all and think about it. They want to be. It tends to be they either dismiss it out of hand initially, or they're dialoguing, and in which case they tend to embrace it. There is one friend of mine, he's become a friend of mine, who's a theistic evolutionist, and I've been dialoguing with him. We're on like round 10 now online, and he read my book. He had me out to give a talk in his community up north of where I live. He's still in the theistic evolution camp, but he's thoughtful. He's read the book. We're dialoguing carefully, so we'll see where it goes.
I'm seeing we're maybe running out of questions, and we're probably good time, but maybe I'll close with a story that's also apropos to your question. man on the screen is named Günter Beckley. I've referred to him a couple times. He's a leading German paleontologist. He's a curator at the the Stuttgart Museum of Natural History, which is the largest natural history museum in Europe. Scores of publications, species named after him. He's an insect guy, insect paleontologist. In 2009, I got an email from him. And he emailed me and he asked me, please do not reply to this email. And he said, you'll also see at the bottom my credentials and my position at the museum, but please do not phone me at the museum. I'd like you to call me at home on this number and to do so between the hours of 7 and 10 German time. So I did. We got in a really interesting conversation. Turns out he illustrates Doug's point. He was at the time not only the curator of a number of different fossil amber and insects and later a Cambrian exhibit, but he was the curator for the Darwin anniversary display in 2009. 2009 was the 200th anniversary of Darwin's birth and the 150th anniversary of the publication of The Origin of Species. Günter had been appointed by his colleagues as the public spokesman for the museum for Darwinism to the media. He created a display to illustrate the strength of Darwinian evolution. It was a scale of justice. On one side was The Origin of Species. On the other were 10 or 12 books on intelligent design. And the scale went like this against the laws of gravity, showing that Darwin outweighed them all. One of his colleagues said, well, look, if you're going to be our spokesman to the media, you better read some of those books so you know what they say, because you might get asked about them. And he said on the phone, that was my mistake. He began to read them, and he said, first he thought, I think these guys are being unfairly maligned. They're not representing their position accurately. This isn't what I've heard about intelligent design. And then he thought, you know, some of these guys have a point, and then he began to get persuaded. And that started an odyssey for him, which ended in 2016 with him publicly announcing that he was an ID proponent, and that led to some other difficulties in his life, like getting erased from Wikipedia. But the point is that, and I think it underscores Doug's point, when we have had people seriously engage the things that we've written in depth, oftentimes they end up being like Günter Beckley. So I thank you all for a really enjoyable night for us and for your rapt attention.